I think the audio's on. All right, we're gonna get started. So obviously your exam's tomorrow. Just make sure, does anyone have questions about like where they need to go, where they are, any of that stuff? Cool. So exam 3 is gonna be on topics like 6 to 8. So that's microtubules and then cell cycle 1, cell cycle 2. So just a little bit about like what we're doing. Oh, I didn't edit this, but the first part of it's gonna be like a more traditional review. So, you know, the slides that you would see either in class or if you come to like weekly review sessions, something along those lines, a little bit more like condensed and not as detailed as the weekly ones, so that we can save a little bit time for the second half, which is going to be more practice questions. There was supposed to be a Kahoot. I didn't get time to make the Kahoot, sorry, but there's still like Kahoot style questions and then like longer questions. So they're all just like mixed together. So some of them are going to be like just content based shorter questions. And then some of them are going to be like longer explanation type of questions so that you guys get a good practice between both of them. All right. And then, yeah, that's it. So we'll go into microtubules first. Microtubules are very versatile and they have a lot of functions. So specifically to talk about what they are, they're polymers. So that means they're made up of tubulin monomers. I think the next slide is going to go into a little bit more about tubulin, but if you see over here, so it's each monomer of tubulin, they come together to make a dimer. So you have your alpha and your beta tubulin, and then a bunch of these tubulin dimers all come together to make a microtubule, which is a polymer. So they have, again, a bunch of different functions, some of them being resisting cellular compression. So that just helps the cells maintain their shape. And then they can help, obviously, during M phase and cell division, pull replicated chromosomes apart. And then they also act as a track for the movement of vesicles. So that's where like your dyneins and your kinesins come in. They walk on the microtubules to carry vesicles, carry cargo, anything that needs to get around in a cell. So there's 3 different types of subgroups of microtubules. You have your aster ones, which I think of like asterisk, which is like the star sign, because it kind of like splays out in a star. So they go from the center at the centrosome all the way to the edges of the cells. And then you also have your interpolar microtubules. They like intertwine with each other. So you go from one centrosome to the other centrosome, and they kind of interlock in the middle. So those are your interpolar ones. And then you have, of course, the kinetochore microtubules, which are these blue ones here. So they go from the centrosome to the kinetochores of the sister chromatids. Those are like the centers at the centromeres, where during anaphase, they can spread apart and like pull the chromosomes apart. And again, yeah, so all of these microtubules are used during spindle assembly and chromosome preparation for the end phase of cell cycle. Also, if anyone has like questions along the way, feel free to just like raise your hand. So again, a little bit more about tubulin. So as I said before, they're made up of 2 monomers. You have your alpha monomer and then you have your beta monomer. The alpha is the like the negative one, and then the beta is the positive subunit. It's not related to charge. It's more related to, you have your plus end of the microtubule and then you have your minus end of the microtubule. So your plus end is where most of the activity, polymerization or depolymerization, is occurring. Theoretically, they can occur on both sides. You can have activity on both the plus and the minus end, but just for like the sake of this class, we say that really all your activities on the plus end, because your minus end is embedded inside the MTOC, which in this case is the centrosome. And then within the centrosome, you have your gamma tubulin ring complexes. So if you want to think of like the centrosome, and then on the centrosome, it's like that picture with like a bunch of holes on the centrosome. So your minus N is embedded within those gamma tubulin ring complexes. And then really important, so tubulin is a GTPase. That means that it uses GTP and GDP. And so only the beta subunit can hydrolyze. So only the beta subunit can go from GTP to GDP. And then something to note as well, obviously, is that when you're GTP bound, the tubulin has good affinity for each other. So it has good lateral affinity and then also good longitudinal affinity. So they can keep that like circular shape and then also build and grow longer. Once you become GDP bound, they don't have good affinity for each other. So that's when depolymerization occurs and they all kind of like fall apart. Okay. So going into polymerization assay in vitro, basically it's like you're doing polymerization in like a test tube. So you're building it from nothing. And so there's 3 specific phases. You have your lag phase, your elongation phase, and then your plateau phase. In the lag phase, so because it's in vitro, you're building it from nothing, it takes a little bit of time for all of the tubulin to come together and even form a seed. And so that's why you have that lag phase. And obviously in that case, then that means if you have more tubulin that are free, you just have like a greater concentration of tubulin, then your lag phase would get shorter because it doesn't take as much effort or it doesn't take as long before your seed forms. And then you can start to build off of that seed and form your microtubule. So then that goes into elongation, where once you have that seed, you're all set. You have like your basis basically. Tubulin can rapidly add to the plus end. And then you have this high absorbance because you're like building and you have just a lot more of this microtubule. But because again, you are in a test tube, it's in a closed environment. It's not like a natural cell where you have an endless amount of GTP. They're like interchanging in and out. You only have a certain concentration of free tubulin, and then you also only have a certain concentration of GTP. It's as however much you put in the beginning. So once you use it up, as you get closer and closer to using it up, your rate of assembly and disassembly starts to become equal. Because remember, as you're adding onto the plus end, you're also hydrolyzing. So once you get to the plateau phase, that's when it's not like it stops. It's not like the microtubule is just still. It's more like your rate of hydrolysis is the same as how much is being added on. Eventually though, again, because it is in a closed environment, if it's in there for a long amount of time, or if it's in there long enough, eventually hydrolysis is going to catch up to the plus end of the microtubule. And again, when hydrolysis catches up, that means everything is GDP bound. There's not a good affinity for each other. So then catastrophe happens and everything just kind of like falls apart. And it won't be able to like rebuild itself because unlike a normal cell, everything is going to stay GDP bound. It's not going to have like the mechanisms inside a test tube to switch the GDP for GTP. So once it's like everything's used up, there's like no going back. You can't remake another microtubule from the same stuff that you originally put in. Does that make sense? Well, any questions so far? So the drugs that affect microtubules, usually on exams and problem sets and things like that, when Laura does ask about these drugs, she'll like put a little parentheses about like what they do. But just in the case that she doesn't, it's good to kind of just know what their function is. So the 3 that we talk about are Taxol, colchicine, and vinblastine. Taxol is a stabilizing microtubule drug. So that means it binds to the microtubule and then it prevents it from depolymerizing. Yeah, so it's not going to disassemble. And then colchicine is the opposite. So it's trying to destabilize microtubules. So it's going to prevent polymerization. And then vinblastine is also a destabilizer. So it's going to prevent polymerization. And for a lot of these, they're used for chemotherapy because we talked about like dynamic instability during class. And that's basically just like the constant building and unbuilding of microtubules, growing and shrinking. because all of that is needed for cell division, as we're going to talk about a little bit later on. But if you don't have enough growing, or if you don't have enough shrinking, or vice versa, then your cell is not going to be able to divide. So that's why they can try to use these drugs to prevent cell division. And then, you know, preventing cell division, you prevent or try to prevent cancer. Okay, so last couple things about microtubules, or in this topic. So we have MAPs, so they stand for microtubule associated proteins. The only map that we really talked about and went into detail about was tau. So tau is a microtubule stabilizer. As I said before, a stabilizer, that just means that it's going to prevent microtubules from depolymerizing. And it's commonly found in neurons in the central nervous system. That's just like a little tidbit. So if you have functional tau, this is how your microtubule looks like. It's stabilizing the microtubule. It's making sure that everything is held together and that it's not going to break apart anytime soon. But if you have a dysfunctional tau and it's not able to do its job, then you kind of have these tangles, is what they're called, because nothing's holding the microtubule together and it's going to start depolymerizing. Think of it as like if you're driving on a highway and then all of a sudden there's an earthquake and the road gives out under you. That's not good. So same thing for like these neurons. The microtubule is the road, and it's just like going to give out if the tau isn't there. And then that means all of the things that are traveling on the neurons, they can't get to where they need to get to. The signals can't get to where they need to get to, like all that. So, oh no, this was supposed to be a GIF. It's not moving. Okay, sorry. The motors are the motors are ATPases. So in microtubules, your tubulin, those are GTPases. Motors are ATPases. So that means that instead of using GTP, they use ATP and ADP, and that's how they function. That's how the motors, like these two legs that like we see a lot, that's how they're able to move on the microtubules. If you haven't watched it yet and you still don't really understand how motors move, in the movie folder 17.7, that's super helpful. I don't usually like recommend movies, but this is the only one I usually recommend. So as a little like breakdown of it, definitely not as good as the movie does, but a little bit of a summary of what it explains is that when motors are bound to ADP, a similar trend when it's hydrolyzed in the like DP form, usually that means you have a lack of affinity. So same for motors, when you're bound to ADP, you have a lack of affinity for the microtubule, so you're not able to bind to the microtubule. As for how it moves, so when the motor makes contact with the microtubule, there's a conformational shift in one of these motor heads, and that makes it so that the ADP diffuses out of the motor head and then ATP diffuses in. So you can think of it as like originally it was floating around, and then the ADP diffuses out, ATP, which is just like in abundance around the cell, comes in, and it's going to swing over because now it has a good affinity and snap onto the microtubule. And then after that, now you have one that's kind of behind the one that just snapped in front. And that one was ATP bound, but it's going to hydrolyze and then become ADP. That way it can let go of the microtubule. And then same thing again, it's going to like snap forward. And then this one's going to snap forward. And it's just like literally like it's walking on the microtubule. The main types of microtubules that we talk about are in eukaryotes, the dyneins and the kinesins. Again, like I said, they move along the microtubules and they can transport, for the most part, that's their purpose. They transport cargo around the cell using the microtubules as like the tracks or the roads. So this is just like a quick little summary of each one. I think if you want like more specific in-depth explanations of them, you can definitely go back into the different reviews, weekly reviews that we've had. But for sake of time, I just did like a little summary for each one. So dyneins are minus end directed. That means they walk towards the centrosomes. So remember the minus ends of the microtubules, those are the ones embedded inside the MTOC. So dyneins walk towards the spindle poles or towards the towards the MTOCs, however you want to think of it. Kinesins are all positive or plus N directed. I think there's like one that we talked about briefly. It's like kinesin 14, that's minus N, but you don't really need to know that. So the ones that are focused on the most are 1, 3, 5, and 13. They're all plus N directed. And for the most part, they're all dimers except for kinesin 5. That one's a tetramer. Tetramer means 4. So you have like 2 dimers. So you have, you have 4 dimers. So like 2 heads on each side and then 2 tails on each side as well. So you have like, this is a better picture. So you have 2 heads on each side and then 2 tails on each side. So they look like dumbbells instead of, in this case, where they're dimers. So you only have 2 and 2. Here you have 4 and 4. But then this is just a picture about each of the ones from the previous slide that kind of gives you like a little bit better of a depiction of how they move and what they look like. Yeah. Another difference between all of them is that kinesin 13 is a little bit different. Rather than so-called like walking, it doesn't really go towards the minus or the plus N. It kind of just stays and like diffuses along the plus N side of the microtubules. And it's a destabilizer. So when it's there, its purpose is to try to like depolymerize the microtubules. An example is MCAC, and then we'll learn about like how that helps move the chromosomes like towards the spindle poles and all that. Any questions about microtubules? Nice.
All right, now we're gonna get into the cell cycle. First is just an overview. So cells start at G1. This is the first gap or growth phase. This is when they prepare for DNA replication. They're growing, they're making sure they have enough nutrients. And during G1, they pass a restriction point if everything looks good. And remember, once they pass the restriction point, they are still in G1. And then G0 is a non-dividing state. So it could be a cell that just went through the cell cycle. It's waiting for another growth factor to start to signal for another cell division. And after G1, we get into S phase. This is DNA replication. There is a checkpoint during S phase as well. If something goes wrong, the cells can stop in that phase. And then next is G2. So the cell is growing again after it has replicated its DNA. It prepares for M phase, which is chromosome segregation. And then there is also a checkpoint for this phase. Next is M phase, which is when the cell divides at the end of M phase at least. It includes prophase, metaphase, anaphase, and telophase. And then there is also an M phase checkpoint. So that's just the overview. We'll get into details, more details in the later slides. First, we'll talk about CDKs. So you've probably heard this a lot. They are the master regulators of the cell cycle. They are serine threonine kinases, which means, so they're kinases, which means they add phosphate groups and specifically on serine or threonines. So they activate protein complexes required for the events in the cell cycle to occur. They are regulated in 3 ways. So cyclin levels, they can only be active when bound to cyclin. Second is phosphorylation or dephosphorylation. And then just remember that phosphorylation doesn't automatically mean activation. We learned about WEE1, which I think there's a slide with more detail about that, but WEE1 adds an inhibitory phosphate. So it's an inhibitory kinase. And then CDKs are also regulated by CDK inhibitors. They can bind to CDK and inhibit it basically if there is an issue during the cell cycle. So the CDK needs both an activating phosphate and cyclin to be able to be active. Yeah, whenever there is whenever there is a CDK inhibitor, it will halt the cell cycle. So it'll put like a break on the cell cycle. And then CDKs target different substrates to activate or deactivate depending on which cell cycle phase it's in. And that depends on the cyclin that it's bound to. So it has to be bound to a specific cyclin during a specific phase in the cell cycle. Cyclins, they give CDK binding specificity. So that goes back to the whole idea that I was talking about in the previous slide, where the CDK has to be bound to a specific cyclin for a specific phase in the cell cycle. So cells produce the cyclin they need to complete the cell cycle phase that they're in. As an example, during M phase, the CDK needs cyclin B specifically. During S phase, CDK needs cyclin A. You don't need to know exactly which cyclin is needed. Mostly you just say like G1 cyclin, but that's just an example. And then if the cyclin's not needed, it is sent to the proteasome for degradation. So cyclins are regulated by transcription and ubiquitin mediated proteolysis. So they're either like created through transcription or destroyed. All right, so then cyclin and CDK together. So just CDK without cyclin, it has an activation loop. And it's kind of tucked inside the CDK and it won't bind to anything unless cyclin is bound to the CDK, which is why CDK needs cyclin in order to be active. So then when CDK is bound to cyclin, it undergoes.
There's people outside and they're saying that they have the room at 7:30 for another review session.
Okay, so where was I? Once CDK is bound to cyclin, it undergoes a conformational change, which exposes the activation loop. And this will allow CDK to phosphorylate other proteins, but it doesn't mean that it's fully active. And then you need a CDK activating kinase or CAC to add a phosphate to CDK's activation loop, which is what completely allows CDK to phosphorylate its target substrate. And then this last point is just talking about the CDK inhibitor. It blocks CDK from phosphorylating target proteins or prevents CDK from binding to substrate. All right. Oh, yes, question. Yeah. I don't think there's a feedback loop specifically with this one. I think it's just that you need both cyclin and an activating phosphate in order for it to be active, and like one of those isn't enough. Does that make sense? Does that answer your question? All right, perfect. All right, so if there is a mutation in the DNA, or if the DNA is damaged, if there is any kind of issue with the DNA, the cell can stop the cell cycle by making a CDK inhibitor. What happens is that P53, it gets activated when the cell senses some kind of issue with the DNA. P53 is a transcription factor that upregulates transcription of the P21 gene. And the P21 gene codes for the P21 protein, and the P21 protein is the CDK inhibitor. So once P21 is made, it can bind to CDK and cyclin complex to inhibit it and pause the cell cycle to allow it to fix whatever issue is going on. And then when P53 isn't needed, it's just degraded. So when it is needed, the cell can make sure it's not degraded so that it can do its job as a transcription factor. Oh, whoops. Yes.
P53, that can happen in all the phases of get those checkpoints when it's checked for like damage.
Yeah, I believe so, yeah. Okay, G1 phase. So some people were confused about the order of events in this phase. I tried to kind of like put things in order here. But basically throughout this phase, the cells are making sure they have enough nutrients, they're big enough, they have enough cellular machinery, so like proteins and enzymes to divide. And then they make sure that the DNA isn't damaged. So early in G1, when CDK levels are low, the pre-RCs form, and then we'll get into more detail about those. Also early in G1, the G1 CDK complexes can still form, but they don't become active yet. So just because they have formed doesn't mean that they're active or that they can inhibit the pre-RC formation. In like mid to late G1, there is the restriction point. This happens after CDK becomes active because CDK needs to be active in order for the cell to pass the restriction point. And then, as I mentioned before, once cells pass the restriction point, they are still in G1 for a little bit. And then in late G1, pretty much right before S phase, the pre-IC forms, which is a complex that builds on the previously formed pre-RC. And then this has to happen when the CDK is active. And yeah, we can get into more detail about that too. So first, we'll talk about the pre-RC. As I said, they form early in G1 when CDK activity is low. The first thing that happens is the origin recognition complex binds to the origin of replication on the DNA. And then that binding can then recruit another protein called CDC6. And then CDC6 and ORC recruit CDT1, another protein, and MCM, which is the helicase. CDT1 is not actually a part of the pre-RC, but it just helps recruit MCM. And then multiple MCMs can be recruited to unwind the DNA in both directions. And then all these proteins together, except CDT1, form the pre-RC. And ORC, CDC6, and MCM are all ATPases. So they can all hydrolyze ATP to ADP. And then CDT is not, but it's also not a part of the pre-RC. Okay, next restriction point. So as you've probably heard, it's the point of no return. It's when the cell commits to the cell cycle. If it passes the restriction point, then it either goes through with the cell division or commits apoptosis. So when the cell is ready to commit to the cell cycle, the G1 cyclin CDKs are active and they phosphorylate a protein called RB. And before RB is bound to E2F and holds it inactive, and once RB is phosphorylated, it lets go of E2F. So E2F is a family of transcription factors that upregulate proteins, or upregulate the transcription of proteins that are needed for S phase. And then once RB is phosphorylated and lets go of E2F, it allows E2F to do that, to act as a transcription factor. And yeah, so once all that occurs, then the cell has committed to the cell cycle. And then the last thing that happens in G1 is the pre-IC formation. So once G1 CDK becomes active, there are more proteins that bind to the pre-RC to form the pre-IC. And CDK is required to get the proteins to bind. So CDK phosphorylates the proteins, which allows them to then bind to the complex. So eventually DNA polymerase gets loaded at the origin of replication and initiates DNA replication. Once that happens, the cell enters S phase. So this class defines S phase as the start of DNA replication. Origin firing is when replication begins from the origin, and then not all the origins of replication along the DNA will fire at the same time, but they can. And then, yeah, next is cell cycle part 2. Does anyone have any questions on what we did so far? All right.
All right, so Iris basically just said all of this. So it's just like entering S phase. You need to make sure that you have all your pre-IC complexes so that it can fire into S phase. And so once you have your SCDKs, so that's because you're in S phase, so you have your SCDKs, then you have to make sure that your pre-RC is not going to be able to assemble again, because pre-RC again is what allows the cell to start replication. If you already replicate or started replication once, you definitely don't want to replicate again, because then the more replication you have, the more likely your probability is for DNA damage, replication errors, mutations, all that jazz. So you definitely do not want the pre-RC again. So the way that the cell makes sure that it's not going to be able to reassemble is by phosphorylating the ORC, phosphorylating the CDC6, and then phosphorylating the MCM. So a little bit about each one. When you phosphorylate the ORC, that prevents CDC6 from binding. So yeah, so it prevents CDC6 from binding. Obviously, if you don't have either of those parts, you're not going to have a pre-RC. To make sure it covers all its bases, it's also going to phosphorylate CDC6 just in case it misses anything. So then like any of the CDC6s from over, just kidding, I don't have a mouse anymore. Oh. So all of the ones from over here that might have been missed perhaps, then it'll be degraded through ubiquitin mediated proteolysis. And so if you don't have CDC6, then you can't recruit MCM. And then again, just in case like this doesn't work, then it's also going to phosphorylate MCM, and then that's just going to kick it out of the nucleus for nuclear export. Just to clarify, so MCM obviously is like, I think it's just like another word for helicase. So it's involved in replication. So all the ones that are not actively participating in replication already, they're going to be exported out if they were not recruited. So yeah, just making sure that like these three main parts are prevented from interacting with each other, because as long as you don't have one part, you're not going to have any of the others, but just the cell's like basically covering all its bases. Okay, so within the S phase, you have your cohesin rings. They form around the replicating DNA. So cohesin rings are rings that keep the DNA strands together throughout and after duplication and into M phase. So the purpose of having them there is so that your chromosomes don't segregate or separate prematurely. So that just ensures that, you know, your chromosomes, you have an equal amount for when you actually do divide. And then another reason is that it has to be present in S phase in order for M phase to be able to occur. So if you don't have cohesin rings, you're not going to be able to go into M phase. So then that kind of goes a little bit into the S phase checkpoint. So the S phase checkpoint is just like the level between S phase and M phase, determining if they can go into that next cell division cycle. So it's checking for if any of the DNA is damaged, if any of the DNA is wrongly replicated, has all of the DNA been replicated? And then if necessary, if it decides that replication needs to stop, or if the cell just has to stop the cycle overall, it could activate P53. P53 is a transcription factor that transcribes for P21, which is a CDK inhibitor. So again, like Ira said in the beginning, CDK is like the master cell regulator. If you inhibit CDK, you're not going to be able to go into any of the other phases. So yeah, so that's why P53 can also be like the signal for programmed cell death. Oops, sorry, I think I said S phase to M phase. I meant S phase to G2 phase. So speaking of G2 phase, this is where the cell is, you know, again, growing and prepping, similar to G1, but it's just that it has to continue growing. This is especially crucial because it's right before M phase. So if your cell is not fully grown, if it's not big enough, if it's too small, if you have damaged DNA and all that, there's not really anything to fix that once it goes into M phase. And then that obviously could lead to like illnesses and cancer and all that. So it's all about in G2 phase, it's trying to work towards M phase CDK activation. If you don't have mCDK activation, you don't have M phase. So everything that happens at the G2 checkpoint leads to mCDK activation. So a little bit about like kind of the first half of it. During G2, your mCDK levels are rising, but it is still inactive because in order for CDK to be active, it has to be bound to cyclin. So in this case, cyclin and CDK bind, but it's still inactive because there's no activating phosphate yet. What happens is that simultaneously you have a WEE1 kinase and then a CAC, which is also another type of kinase. WEE1 is inhibitory, CAC is activating. So they're both going to put on phosphates onto the mCDK, but they're going to be two different phosphates. So one's inhibitory from WEE1, and then one is activating from CAC. So even though both of them are on there, the inhibitory one is stronger. So as long as this one is on the mCDK, it's always going to win, if you want to like think about it that way. As long as your inhibitory phosphate is on the mCDK, it's not going to be active. So you're not going to go into M phase. And the purpose of having this inhibitory one is because, again, you're in G2. So you don't want to immediately have active mCDK and immediately go into M phase, because otherwise your cell is going to be too small. It's not going to have grown enough. There wasn't enough time for it to check for any damage. So the purpose of having the inhibitory is so that the cell gets a little bit more time to do what it needs to do in G2. From there, once it feels like it's all set, that it's ready to move on, CDC25, which was partially activated by PLK1, which is another kinase, it's going to go and take the inhibitory phosphate off. So CDC25 is a phosphatase. It's going to take off the inhibitory phosphate specifically. So once you take off the inhibitory phosphate, all you have left is the activating phosphate. So now your MCDK is officially active, but it's still not fully like to its full capacity. So you want to get into M phase as fast as possible. So what happens is that a positive feedback loop occurs. Once you have this active MCDK, it's going to go back and further activate what was like this. Originally, the CDC25 was partially active. So now it's going to be fully active. And this whole process, like this loop over here, it's just going to happen faster. So you're going to have more MCDK active so that you can get into M phase quicker. So this is just the M phase overview. I won't go too far into it because we're going to talk about each one a little bit. But if you like ever want like a little summary, that's what this is for. So first you have prophase. Prophase is when the first part of M phase where your active MCDKs that we were just talking about, they are doing their job. So what they do is they phosphorylate a bunch of things. They phosphorylate lamins, which is what breaks down the nuclear lamina or like the nuclear envelope, however you want to think of it. And then the phosphorylate, it'll also phosphorylate condensin, which is what causes these chromosomes, chromosome condensation. So they go from being like squiggly in like all those pictures that you see to being more like condensed together and they look like sausages, I guess. I don't know where this came from, but it just stuck in my mind. So I always put it there. And so another last thing that they phosphorylate are different maps and motors that we were talking about before during like the microtubule section. And they just help with mitotic spindle assembly, which is really important for like the spindle assembly checkpoint. So some more main ideas that I kind of compiled all together. So again, if you want more like details about each one, if you go into the different like weekly reviews, there's definitely more detail there. But just to bring across the main ideas, you have sequential phosphorylation. And this is just the idea that phosphorylation has to occur in a specific order. If something upstream has to be phosphorylated, but it's not, everything downstream can't occur, and vice versa. So that's also why CDK is a master cell regulator, because CDK is a kinase, it phosphorylates a bunch of things. So if you get rid of CDK, maybe it won't phosphorylate what it was supposed to. So then everything after that isn't going to happen. So if you want to cut off like S phase, maybe you'll cut off the SCDK and then whatever needs to be phosphorylated won't be phosphorylated, and then so on and so forth. Your cell cycle just is going to stop. There's also spindle assembly. So this is when your existing microtubules depolymerize. So in a cell in general, you have microtubules, they offer structure for the cell. When you start to go into M phase, all of those microtubules that were already formed and already existing, they're going to depolymerize and basically just like disassemble. An example that some like a protein that you could use is catanin. And basically what it does is like it cuts up all those microtubules so that they can again quickly reassemble and like repolymerize into spindles so that they can prepare for chromosomes. There's a little bit more about spindle assembly specifically later on. But connected to spindles is your kinetochores. They are large protein complexes at the centers of the sister chromatins. That's where specifically spindle microtubules attach to. And then also opposing forces. So I know like a lot of people get confused about this, but opposing forces are what are required in order for spindles to be stable. So you have like basically push and pull where the spindles are like pushing and pulling against each other using kinesin and dyneins because they're moving in opposite directions in order to stabilize and offer that tension within a cell. So this is just kind of expanding on the push and pull model. For example, your white motors, like this one, it's a dynein, so it's minus end directed. So essentially what it's doing is it's trying to walk towards the minus end, so towards the centrosome. And as it's walking towards this way, it's basically like dragging the spindle behind it. So that's why the motion of this spindle pole is going to be pulled outwards, whereas maybe some of the positive end ones, they're going in the opposite direction. So you have like this push and pull idea where like you're causing a lot of tension inside the cell. Yeah, and then like I'm gonna send out these slides later on. So in like the notes section of Google Slides, there's like a little bit more about each type of motors that are on this picture if you want to understand it a little bit more. Okay. So going into metaphase, this is where your sister chromatids are still secured by cohesin rings. So they're still combined together or connected together. And they are attached to microtubules at the kinetochore. It doesn't show it here, but pretend there's a kinetochore and they're connected to microtubules. What happens for metaphase is that, M for middle, so they all line up in the middle, and the equal and opposing pulling forces from that push and pull model, it brings all the sister chromatids to the middle, and then the cell can basically like sense that tension, that it's ready and that they're all lined up and that they're ready to actually separate now. So in order to separate, it has to get rid of those cohesin rings so that they're not like bound together anymore. And that's what the spindle assembly checkpoint is all about. So you have your CDC20, and it's going to go bind and activate APC, which is a ubiquitin ligase. So it's going to go and degrade securin. So beforehand, securin and separase were bound together, but once APC is activated, it's going to go and degrade securin. If securin's out of the picture, now separase is free to do whatever it wants on its own. It's going to have a conformational shift and then go and basically cut the cohesin rings. And so now that the cohesin rings aren't bound or aren't binding the sister chromatids together, anaphase can happen because there's nothing stopping them from like keeping them together. So anaphase, we're almost at the end of M phase. Anaphase is when you separate the sister chromatids, right? So they get pulled apart to their respective spindle pole size of the cell. There's two different types of anaphases that can occur. You can have anaphase A or anaphase B. A is all about the chromosomes being pulled poleward, so like from the middle to the edge, whereas anaphase B is about the poles being pushed apart, so from edge and even further out. So A, I don't really have like a good way to remember this, but A is basically from the middle and you're going outward. B is also outwards, but you're going like you're already at the outside edges and you're just going even further out. And then these are kind of the different ways that A can occur and B can occur. So if your chromosomes are pulled poleward, that's of course A, because your chromosomes are pulled poleward. They can be done so by dyneins, so negative directed motors. If you have your dyneins like at like the kinetochores, they can walk towards the minus end and basically like chew back on the microtubules and shorten them so that as this gets shorter, the chromosome's gonna like move closer towards the spindle pole. Yeah, so kinetochore microtubules can shorten, and then if the spindle poles are still in their place, that would also be A, because as soon as you start moving the spindle poles, that's going to be anaphase B, which is, yep. So again, like your poles can move outside, your spindles can lengthen. This is more like kind of like contradicting to think, I guess, because you're thinking about the spindle, so you might think that it's A. But if you think about it, if your spindle poles are lengthening, if your spindles themselves are lengthening, then there's like more room for, or there's less room than there originally was for the spindle poles. So in order to make more room for like this lengthier pole or for this lengthier spindle, they have to move outward so that not everything is like squished together. Does that make sense? I know sometimes A and B is like confusing to distinguish. Cool. So, okay, so last part is telophase and cytokinesis. It's talked about the least in class, I think, because there's not too much to it. It's when your sister chromatids after anaphase A or B or a combination, which is usually the case, they finally reach their opposing poles and you have a cleavage furrow start to form. And the cleavage furrow is formed because of a contractile ring that like deepens and deepens and like constricts the cleavage furrow until eventually they touch each other or they hit the spindles that are like whatever spindles are left in between. And then that's basically just gonna like split the two cells apart into two different ones. As kind of, so that's like the end of telophase and cytokinesis. But as a reminder, the cell cycle, it's called a cycle for a reason, right? So when you reach the end of the cycle, your M cyclin CDK levels have dropped. And that means your spindle disassembles, your chromosomes can start to decondense again. So now you're going from sausages back to spaghettis. And then your nuclear envelope or that nuclear lamin starts to reform around the DNA again. And then you have two separate cells in their own entirety. And then it can restart everything that we just talked about, because when you have low CDK, your pre-RCs can form. And if your pre-RCs can form, you can start to fire replication again. So that's kind of how like the cycle comes full circle. After this is like end of content and more into practice questions. Does anyone have questions about contents?
I thought that that was like sequential, like or is it like either or?
So I'm positive that both happen. You need both in order for anaphase as a whole to occur. I'm not sure if it is like A happens first and B happens first, or if like the cell's just wishy-washy with that.
It depends
you
on the
know?
type of cell.
Oh, okay.
So it can go B first and then A, or it can happen at the same time, or like it can happen in different orders, but it depends on the cell specifically.
But both definitely do occur. Yeah. Anything else?
All right, now we're gonna get into some practice questions. Some of them are like more problem set type that you might see on an exam. Some of them are like more simple, but our first question. Did I skip? Okay, I did skip one. All right, so which of the following are properties of microtubules? Explain yes or no, why or why not for each.
Would you guys prefer like having a few minutes to think about it yourself, or if we just like go one by one? Okay, votes for a few minutes by yourself. Okay, and then votes for one by one. Okay, a few minutes by yourself.
So there's another review session supposed to be this year?
Yeah, so this, I'll just. Sure, one of the TAs for that class though, he was in my bio tab here,
Really?
but I didn't know him that well, so I didn't want to be like say his name and it
Oh.
not be him. So,
That's funny. Well, he didn't say your name either.
yeah, exactly. We can go over it.
Alright.
Like start with A and then.
Wait, after A, give one more time or no?
Oh, just like, yeah, just go
Just to
through
go
all of
through
them.
them.
Yeah.
Alright. Do you want to unmute?
Oh, I think, yeah, I did.
Oh, cool. All right, we're gonna go over these. So the first one, it says, microtubules are composed of a heterodimer of alpha and beta tubulin. Does anyone think they know the answer to this? Yes. Can you explain? Right, so tubulin, yeah, no, you're right, you did a good job. The answer is yes. So tubulin is the monomer of microtubules. It is a heterodimer, which means it's composed of two different subunits, the alpha and beta subunit. They are different proteins and have a high affinity for each other, which means they don't dissociate. Next one, tubulin has the ability to hydrolyze ATP. I'm seeing some head shakes. So yeah, you're correct in saying no. Tubulin is a GTPase, which means that it can bind to GTP and hydrolyze it, but that says nothing about ATP because GTPases can't hydrolyze ATP. Okay, next one. A good analogy for microtubules is that they are like cars on the road transporting people where they need to go. Yes. Yes, that is correct. So the microtubule is the road, and cargo is transported like through the road or using the road. The motor proteins are considered the cars because they bind to cargo and walk along the microtubule, bringing the cargo along with it. All right, next one. Microtubules are unstable. Can I get like a thumbs up or a thumbs down? Yes, okay, seeing a lot of thumbs up, that's correct. They're naturally unstable because they have the ability to hydrolyze GTP, and when they do, they depolymerize. And there are ways to stabilize them if necessary. And then next one, microtubules are hollow. Pretty simple, thumbs up or thumbs down. Yes, they are hollow. They're made up of a protofilament ring, which is 13 protofilaments that interact laterally to form a cylindrical structure. And then there is a space in the middle of the cylinder, so it is hollow. Last one, microtubules often extend out from an organizing center in the cell called a centromere. Thumbs up or thumbs down? Okay, I'm seeing some mixed reviews. This is actually false. Microtubules do not extend from a centromere, they extend from the centrosome. So they are two words that sound very similar but mean different things. Centrosomes are microtubule organizing centers in animal cells. The centromeres are what we talked about with chromosomes, where the kinetochore builds off of.
So centromeres are the places where like the sister chromatids are connected in that middle place.
Right. All right, does that make sense to everyone? Any questions? All right, next question.
So dynamic instability causes microtubules either to grow or to shrink rapidly. Consider an individual microtubule that's in its shrinking phase. So for both for all of A, B, and C, think about it from its shrinking phase. I think this one we could do like A and then a little bit.
Yeah. Makes sense.
If anyone has an idea for what they think the answer to A might be, you can like feel free to raise your hand whenever. Does anyone have an idea for what might cause a microtubule to go from shrinking to growing? Yeah, go for it. Specifically, what about, like, it should be, there's like a little bit something else to it.
It should be greater than. Right,
Yep, so exactly. So just like tweaking your words a little bit. In order for it to stop shrinking and start growing, you want to have more tubulin, but more specifically, more GTP bound tubulin, or it can be, yeah, so GTP bound tubulin, as long as that rate is higher than the hydrolysis rate or shrinking, then it can basically like make up for it shrinking. And the GTP will like overpower the GDP and then it'll start to grow again. Nice. How would an increase in the tubulin concentration affect this switch from shrinking to growing? If you think that the switch from shrinking to growing will switch faster, raise your hand. If you think that switching will occur slower. Okay, I'll just explain it because so similar to part A, in order to go from shrinking to growing, you want to have more GTP around, right? Because that's where you have more affinity for each other. If you have a lot more tubulin that are all GTP bound, then you'll be able to go from shrinking to growing much faster because you just have more of it around so that it'll like overwhelm the microtubule that's shrinking faster. So yeah, so it's just like the more you have of it, the faster it'll occur because there's more of it around. And then what would happen if GDP but not GTP were present in the solution? Oh, go for it. Yeah, it wouldn't be able to grow. Nice.
All right, so let's see this one. Kyra, an undergrad student taking Bio 285, decides to pursue her fascination with the microtubule cytoskeleton by taking on a new research project. She plans on studying microtubule formation and dynamics, both in vivo and in vitro. Being the eager researcher that she is, she wakes up on Monday morning to start her first experiment. She adds GTP and purified tubulin in a test tube and observes microtubule polymerization in vitro. She takes a few time points up until 11 a.m. and observes the following. So that's the graph that she gets. And then explain Kyra's results at the molecular level, what is happening inside her test tube over time, and what do the stages numbered above represent? All right, any ideas for this question? Any ideas for what the numbers are representing or what's going on in the test tube? Okay, so over time, the tubulin will bind to GTP and polymerize, resulting in the increase that you see on the graph of the amount of microtubule polymers present over time. The section that's labeled 1 is the lag phase, which is when tubulin dimers form oligomers and then form the protofilaments. And then those protofilaments will then interact with each other laterally to make that hollow tube-like structure. And then it takes time for that process to occur, which is why the amount of polymer that you see doesn't increase right away. It takes time. And then the second, or the section labeled 2 is the elongation phase, which is when more GTP bound tubulin is added to the microtubule, and then that causes the microtubule to grow at a constant rate. So that's what you're seeing with that like increase in number 2. And then this question has a couple more parts to it. Okay. Nope. Okay. Part B, if Kyra had more time to take additional time points later on, how would you expect the shape of the curve to change? Yes. Yeah, perfect. And then would Kyra's results differ if she had used ATP instead of GTP?
Yeah, why?
Yes. All right, the next morning, Kyra repeats the experiment, but she begins the experiment by adding small microtubule seeds at the start of the experiment. Explain how the graph of the amount of polymer versus time would be different from the original graph.
Can you explain why? Yeah, exactly. Just so like the Zoom hears it was as well. When you have more concentration of anything, again, it just like speeds up the process because you have a greater concentration, there's more of it around. So that phase entirely is just going to be shorter. Nice. Okay, these next two questions are also about microtubules, but they're a lot shorter. And then we'll go into cell cycle questions. Okay. You have a polymerizing microtubule and then decide to cut it down the middle. What's your new plus end going to look like? Exactly. So for the most part, I think this is a pretty common question that Laura asked, but so you guys have probably like seen it before, but it's just good to know that like throughout your entire microtubule, hydrolysis is like the majority of it. It's just that GTP bound is a lot stronger, so that's what's keeping it all together. But once you cut down the middle, that's all GDP bound, so it's gonna like depolymerize.
All right, you perform a microtubule polymerization assay with purified tubulin, GTP, and tau. How would the expected graph look like compared to a normal assay without tau?
Yeah, go for it.
Yeah.
Can you explain?
There's,
Right. So just so that the recording hears, tau is a microtubule stabilizing protein. It binds to existing microtubules and prevents depolymerization. But because there is a finite amount in a test tube, the plateau will still be reached, but just later.
I just wanted to clarify on what you said too. So the rate of elongation doesn't necessarily increase. So like if you compare like the two original versus like this type of graph, the slope would still look the same. It's just that the elongation will go longer than usual because you're not depolymerizing or you're not like hydrolyzing as much behind it. So yeah, so like the amount of tubulin that's being added on is still about the same. It's just gonna be added on like longer. So yeah, just like that distinction. So starting into the cell cycle questions, it's been shown that HPV can cause cervical cancer. And in fact, there's now a vaccine for HPV for this reason. The virus encodes a protein called E6, which has been shown to bind human P53 protein. Based on this information and what you know about P53, what effect do you think viral E6 protein binding is going to have on P53? So like, yeah, take a minute to like think about it. Yeah, go for it.
I have a question. So is it that 53 is always translated, transcribed, and degraded, like it's always the cycle of that, and then when it's needed, it doesn't get degraded? Or is it only produced when it's needed?
I think it's usually produced when it's needed.
I think it's degraded.
Oh, it's,
I think it's degraded when it's not needed. Yeah.
wait, what's the difference between produced when needed and degraded when not needed?
Well, so it would always be produced,
Okay, I got you.
But-
yeah.
degraded if there is no issue with the DNA. Yeah.
Russ, I thought that, isn't it like if there's DNA damage, if there is DNA damage, doesn't like keep it, like, doesn't somehow get phosphorylated, so it doesn't degrade, so. Minus the P21. And like, you know, can it still bind to P21 once it's once E6 is there, or is that is just it is binding with P21?
So P53 is a transcription factor. It's not going to be like phosphorylating or things like that. So it's going to, so P53 is going to transcribe P21. And then, so in this case, like knowing that aspect, and then thinking about like what P21 does and what P21 is, how would E6 affect that? That's kind of what the question's asking. Yeah, if that helps. Does anyone want to take a stab at it, or do you want to like think about it a little more? Yeah, go for it. Yeah, exactly. Nice. So it's telling you that the HPV virus encodes for E6, and then that could lead to cancer. So that's telling you that E6 is like doing something that's allowing the cells to constantly divide and go through the cell cycle essentially. So if P53 is usually what causes P21 to be activated, and then P21 is a CDK inhibitor. So P53 is what would allow cells to stop the cell cycle or go through like apoptosis or anything like that. That's like a signal for ending the cell cycle. So if E6 is causing cancer, there's just a lot of the cycle happening. So E6 is probably preventing P53 from doing its job so that it can continue to go through the cell cycle, even if it's not supposed to. Does that make sense?
Just a side note, this whole idea of like cancerous phenotype and like tumor suppressors, proto-oncogenes is like pretty classic like LoRa topic. You guys have a question on your problem set that lists a bunch of proteins in the cell cycle, and it asks if it's a tumor suppressor proto-oncogene. That was on my previous test, so that's definitely like a test-worthy question. And I wouldn't be surprised if you see something that has to do with that topic on the exam. So just in general, a cancerous phenotype or something that is a proto-oncogene will, it will be something that will progress the cell through the cell cycle. It's something that's a tumor suppressor, it will be a protein that its job has to do with pausing the cell in the cell cycle. All right, next question. What needs to be released in order to make it past the restriction point? Yep. Yep. Pretty simple. RB holds E2F inactive. Once RB is phosphorylated by CDK, once it becomes active, it can release E2F, and then E2F can do its job as a transcription factor that upregulates transcription of S phase proteins. Yeah.
Is it like less cycling that binds to the CDK?
It would be G1 cyclin because this whole process is in order for the cell to pass the cell or pass the restriction point, and that's in G1.
S-cyclin and S-CDK, that's for all of the like phosphorylations to get rid of like the pre-RC things. That's where that's comes in play.
Mhm.
And then kind of like relating it to the previous question. So in this case, like E2F would be more of like a proto-oncogene type of phenotype, because by allowing E2F to be released, you're moving on with the cell cycle. So if you like prevent E2F from doing its job, then you're going to stop the cell cycle. Another just simple, like shorter question. What cannot be formed again once CDK is active? Yeah.
Yep.
Nice. Yeah, so I just said again, like specifically like the SCDK. Yeah, once the SCDK is active, it's gonna go like phosphorylate all those different pre-RC port parts. So like the ORC, MCM, and CDC6. And then if those are phosphorylated, they can't come together and you can't form the pre-RC.
And remember CDK is inhibitory to pre-RC, but necessary for pre-IC. Okay. All right. So in the class, we discussed ways in which CDK activity is regulated. The figure you see to the right shows the changes in the level dotted line of M phase cyclin and activity solid line of M phase cyclin CDK during the cell cycle. So for each of the following, determine if they are true or false with respect to how cyclin levels are controlled and how M phase cyclin CDK activity is controlled, and explain your reasoning. So the first one, increase in cyclin levels is controlled at the transcriptional level. All right, any ideas for this one? I'm seeing thumbs up. Yes, so that's true. Cyclin levels increase when transcription of their gene is upregulated, and their levels decrease when they're degraded. When expression of cyclin is increased, there are more cyclin CDKs in the cell. And then the second one says all M phase cyclin CDK complexes are activated once they exist in the nucleus. I see a thumbs down. Yes, so this is false. So CDK has to be phosphorylated by CAC, the CDK activating kinase, and it has to lose its inhibitory phosphate put on there by WEE1 in order to become active. Even when M cyclin is bound, as long as it has the inhibitory phosphate, on it, but by V1, it will still not be active. And then number 3 says cyclins are kinases. All right, I'm getting a general no, which is correct. CDKs have kinase activity, and cyclins simply bind to CDK and give it binding specificity, but they are not kinases. And then the last one, it says a negative feedback loop causes rapid activation of M phase cyclin CDK kinase activity. Yeah, okay, I see a lot of thumbs down. That's correct. Well, you're right in saying no. So this statement is false. CDC25 is partially activated by another kinase, which then allows it to activate CDK. And then once CDK is activated, it can phosphorylate and then further activate CDC25, which allows it to continue activating CDKs. And then active CDK can also phosphorylate V1 and inhibit it, which prevents V1 from adding that inhibitory phosphate on CDK. So this is a positive feedback loop, not a negative feedback loop, because activation of CDK leads to activation of more CDKs, which is why the CDK can increase so rapidly.
Negative feedback loop would be like if it makes less of it or like stops it.
And then why doesn't cyclin CDK activity rise concurrently with the rise in cyclin protein levels? Right. So yeah, when the cyclins are made and bind to CDK, they're not activated right away because of that inhibitory phosphate. And it has to be phosphorylated by CAC and dephosphorylated by the dephosphorylated the inhibitory phosphate has to be dephosphorylated and it has to be phosphorylated by CAC in order to become completely active. And then the last one says, besides the mechanisms discussed, illustrated, or asked about in this question, what is one other way that CDK activity can be controlled in the cell? Yep. Yeah, CDK inhibitors are one. It can be degraded. Yeah.
Okay, moving along, I don't think there's too many left, but what might the consequences be for the cell if CDC25 has a loss of function mutation? Would cells enter M phase earlier or later than normal? Might cells be bigger than normal or smaller than normal? And then we could do like thumbs up for early. Oh, actually, you can go answer it.
It would be something you do for a while because that support makes it like getting past the checkpoint.
Yeah, exactly. And then specifically, so again, it'll be stuck in G2, but more specifically because CDC25, I don't know if you said it, but it's in charge of like taking off that inhibitory phosphate. So if it can't take off the inhibitory phosphate, it's the cyclin CDK is going to remain inactive, either indefinitely or maybe just later than normal, because the cell, like whatever happens in the cell, it's not definite as usual. And so in that case, would cells be bigger than normal or smaller than normal if it's stuck in G2 for longer? Okay. Yeah, so bigger. because it's stuck in G2 for a little longer, and then it's going to grow for longer as well. On the flip side, what if we1 had a loss of function instead? Would cells be going into M phase earlier or going into M phase later? Yeah, and then would it be bigger or smaller? Nice, you guys got it. Because we1 is inhibitory, so if it's not doing its job, it's not inhibiting, M phase cyclin is active, it's going to go into M phase quicker. It didn't have enough time to grow, so it's going to be smaller. Awesome. Yeah.
So can it still enter M5 phase without CDC25?
If we1, are you asking about part B?
A, the first part. So you said it'd be bigger than normal, would it still enter M phase at that point? Or?
Probably not. But like as Laura usually says too, like we don't know, we're not in the cell, so we don't know like 100% sure about everything. But probably not. It's just going to be bigger because like it's going to be in G2, so it's going to be like growing for longer. Does that make sense?
Yeah.
All right. Based on your understanding of the regulation of the eukaryotic cell cycle, how could you explain each of the following experimental observations? The first one says, when purified S-cyclin CDK complex is injected into cells that have just finished dividing and are entering G1 phase, pre-RCs do not form on origins of replication, and the cells never enter S phase. Yeah. Exactly. That S-cyclin CDK is gonna phosphorylate ORC and CDC6 and MCM,
Yeah.
right? Those three proteins. And then the pre-RC will not be able to form. Next one, when a mutated M-cyclin that can't be degraded is introduced into cells, they enter into M phase but cannot emerge from it and cannot re-enter G1 phase. Yeah. Right. So M-cyclin, it does a couple things. So it breaks down the nuclear lamina. It
Condenses,
condenses the chromosomes and
phosphorylates a bunch of other things.
yeah, it does a bunch of things that basically helps the cell begin to like prepare for division. So if it can't be degraded, then the cell will not be able to leave M phase.
Yeah, like you won't be able, because in order to leave M phase, you want to make sure you get your nucleus back essentially. So if you don't degrade your M-cyclin, you're not going to get your nuclear envelope back, and then your chromosomes are still going to remain condensed instead of decondensed. Yeah.
Right. And then last one, mutations that inactivate the main protein phosphatase used to catalyze protein dephosphorylations later in M phase cause a long delay in the reconstruction of the nuclear envelope that normally takes place at the end of M phase. Any ideas about this one? So this one is similar to what we talked about, the last one. If a mutation causes the phosphatase that inactivates MCDK to be inactive, then MCDK will continue to be active. And since MCDKs cause nuclear envelope breakdown, the nuclear envelope would continue to break down and it would not be able to reform. Yeah. What? Could you repeat your question?
Is he just pretty much saying that he is considered?
Oh,
So I think it's saying that for M phase cyclins, in order for them to, so in order for M cyclins to do what it needs to do at the end, like lower their levels, they have to be dephosphorylated. And then it's inactivating the phosphatase that's causing dephosphorylation. Yeah, does that make sense? Okay. Yeah, the word, it's always the wording that's a little tricky. So that's why we try to like give that extra practice. Okay. Any other questions for this one? All right, last question. Last question. If M phase CDK is just kidding. I guess this is similar to one we already did, but if M phase CDK is activated too early, what happens? I heard someone over there say it. You want to say the answer? Yay. Okay. Yeah. So M phase CDK is activated too early. So that means it's not going to be it wasn't stuck in G2 long enough, basically. So it didn't have enough time to grow and it's going to be too small. The next slide, I think it's just the thank you slide. But that was the last question we had. So does anyone have any like last remaining questions of anything? All right. In that case, thank you for coming. If you have any like following questions, you can email us or anything. Feel free to take more donuts, please. I don't want them.
good luck on your exam tomorrow.
Oh yeah. Good luck, guys.